Okay, hello everybody. Thanks for being here so quickly after lunch. My name is Shelley Sianta, and today I'm going to share some pretty fresh data with you all, focused on investigating the scales over which introgression varies in a system that's at the later stages of speciation. So we know that speciation is the process by which reproductive isolation evolves, and undoubtedly that happens most easily in allopatry, where the homogenizing effects of gene flow can't swamp out divergence. But we know that ranges move and species can come back into secondary contact. It's been famously said that secondary contact, or that sympatry upon secondary contact, is the final test of speciation. It's this arena where we get to find out if the reproductive barriers that evolved in allopatry are strong enough to isolate species. If they aren't, we could get a hybrid swarm, or even if there's fairly complete reproductive isolation, there can still be some hybridization and introgression. And with the increase in genomic tools, we know that introgression is a fairly common phenomena at both contemporary and ancient timescales. And one take home from this work, as we've been seeing a lot in this conference, is that hybridization and its outcomes are highly variable. So in addition to thinking about whether introgression is happening or not, some of the exciting questions circle around how and why introgression varies. And we can think about this across different scales. For example, say we have this zone of secondary contact shown here by the overlap in these two species ranges, where there are multiple sites at which species co-occur. We can think about variation in terms of whether introgression is asymmetric between taxa, for example, with more gene flow from one taxon to the other. We can also think about how introgression varies across space. So in this case, among these coarse spatial scales relative to dispersal, so among these replicate contact zones. And we might expect magnitudes of introgression to vary at this spatial scale if populations have differences in their demographic histories, or if environmental features at these sites differ in ways that mediate reproductive isolation and selection on hybrids. And we can also think about how introgression varies at more fine-grained spatial scales, so within these zones of contact where taxa are interacting. And depending on the distribution of parental taxa in the landscape and whether they're partitioned along these environmental gradients or in discrete microhabitats, we might imagine that the degree to which an individual is admixed is not random on the landscape. For example, we might expect to see individuals with high levels of admixture, like early generation hybrids, in intermediate ecological environments relative to the two parents, or even without some underlying environmental feature, we might expect to see highest admixture where these taxa are proximate to one another, just as a function of the balance between dispersal and selection. So we've been studying the scales over which introgression varies in a California wildflower, Clarkia xanthana. This is a species endemic to the southern Sierra Nevada foothills of California, which is a region with really high variation in topography, climate, and ecosystems. The species is comprised of two taxonomic subspecies that differ in their mating system and have diverged recently, about 65,000 years ago. And while these subspecies are primarily allopatric and generally occur in really different habitats in their primary ranges, with the sulfur in more xeric habitats than the outcrosser, the ranges do overlap in this narrow north-south region, which is only about 5 by 50 kilometers long. And within this region of sympatry, these taxa co-occur at these discrete sites where individuals of either taxon can grow within meters of one another. So studies in the field and greenhouse have shown that the taxa are largely isolated by multiple prezygotic barriers like flowering time, and they can form fertile hybrids. And so to better understand the history of divergence and introgression across these contact zones, we did a project where we sequenced whole genomes of 200 individuals across the range of each taxon and in 6 of these contact zone sites. And what we found was that introgression is highly variable. So in this graph here, I'm showing you admixture proportion on the y-axis for sulfurs in orange and outcrossers in blue. You can see that at any given site, introgression is largely asymmetric with more introgression into the outcrosser. And that across these sites, introgression into the outcrosser is highly variable. We used site-specific climate data that was collected over the past 15 years to see if the environment across these sites explained this variation, and did find that one variable, interannual variance in spring precipitation, largely predicted the magnitude of admixture proportion in the outcrosser. And I'm happy to talk about this more later, but we think this is tied to the way that variance across years in spring precip will allow for more overlap in flowering time in any given year, increasing the opportunity for hybridization. And so this variation across sites is super cool, but we only sampled about 7 to 10 individuals per taxon per site for this analysis, so we don't really have a great sense of what's going on within each contact zone. Specifically, we want to know how these zones are structured. So if you take all the individuals, line them up by ancestry, do we see 2 modes in this distribution reflective of largely parental genotypes? Or do we see a scenario with, say, parents and early generation hybrids? And second, we want to know if this variation in introgression is partitioned at these fine spatial scales within contact zones. And this might be the case because the parents themselves are spatially structured. So here I'm showing you GPS individuals represented by the plus signs here with buffers around each one, with the outcrosser in blue and sulfur in orange. And what you can see is that, they're largely separate. And Lauren Carley, a former postdoc in our lab, found that that is statistically the case relative to chance. And given the habitat differences in allopatry and personal observations in the field, we think it's likely that these taxa are partitioned along different microhabitats. So when we think about how the degree of introgression among individuals might be spatially structured, we might predict that admixture proportions would be greatest in regions where where the taxon boundaries overlap or abut one another, due to both the greater chances of heterospecific gene flow there and possibly more favorable selective landscapes for early gen hybrids if these boundaries really reflect these microhabitat differences. So to start answering these questions, we densely sampled individuals within 4 of these contact zone sites, where here each pin represents an individual, again, sulfurs in orange and outcrossers in blue. And we really try to cover the extent of each taxon's distribution without sampling too much locally. We sampled an average of 50 individuals per taxon per site. 3 of these sites, S22, SAW, and SM, were in that previous whole genome study, and they represent a lot of this among site variation and introgression shown in this plot on the top left. But we also included this 4th site at S6, which is new to us and is interesting for a couple of reasons. It's at the northernmost range for both of the subspecies, and it also has some really weird floral variation in that population, and there is some documented variation in cytotypes there. So for all of these individuals flagged on the map, we collected seeds in the field, grew out these plants in the greenhouse to phenotype them, and collected tissue for GBS, and then used hidden Markov models to call ancestry across the genome, using allopatric populations from that past study as our reference panel. So the first thing we did was took all these individuals, sorted them by ancestry to ask what the structure of these contact zones were. And we find that all 4 of them are largely bimodal. So this histogram on the right here is showing the amount of sulfur ancestry in the genome, with the individuals of the outcrosser primarily on the left here, with low sulfur ancestry, individuals that are sulfur-like on the right side of the graph. And as you can see, we have no individuals falling out in the middle. So a few things to note here are that with the inclusion of this S6 site, we continue to see these increased levels of introgression in the outcrossers. That distribution is further to the right on that axis. And we also see the highest levels of introgression into the sulfur at this site. And lastly, we see some more variation in introgression within the sulfur than we did in this past study, as seen by these couple peaks here at this S22 and a little bit more variation in this distribution at SM. And I just want to hop on a quick tangent for a second to say that this new S6 site is really weird, but in kind of a few good ways, or exciting ways at least. So not only does it have the highest admixture proportions of all of these sites, but the 2 taxa at this site also have more similar flower size relative to the other sites. So the sulfur has marginally larger flowers, the outcross has smaller flowers than these other sites, with more overlap between the 2. And we think that flower size is important for speciation in this system, and I don't want to give any spoilers, but you guys should stay here for the next talk to find out why. And then lastly, from this past whole genome resequencing project, we had found some evidence of chloroplast capture where some outcrosser individuals in the contact zone had the sulfur chloroplast. And we had an undergraduate researcher in our lab, Amal Suri, design taxon-specific primers for the chloroplast, and she's been working on amplifying those out of all of these individuals. And what she's been finding is that in the sulfur, basically all the individuals have the sulfur chloroplast. And in the 3 initial populations we surveyed before, the outcrossers mostly have the outcrosser chloroplast. There are some outcrossers with the sulfur one. But at site 6, all of the outcrossers have the sulfur chloroplast, which is weird and cool and would, potentially suggest some sort of different demographic history in this population, maybe hybridization followed by a bottleneck, and it's something we're excited to follow up on moving forward. So we also wanted to know if there's this predicted negative relationship between admixture proportion and distance to heterospecific neighbors. So as a test of this, we took every individual, calculated the average distance to the closest 3 heterospecific neighbors, and determined if that distance predicted admixture in a linear regression framework. And what we find is that largely in the majority of these taxon-contact zone combinations, there's no relationship between these 2 variables, except in the sulfurs at S22. And interestingly, this pattern is largely driven by these individuals down here with basically no admixture in them. And when we look at clustering analyses, these fall out into these 3 different clusters that are also clustered on the landscape, like all kind of relatively right next to one another, suggesting that they're like these inbred families that might, be drifted relative to one another and might have efficiently purged out their introgressed ancestry. Okay, so to wrap this up, we find that with greater sampling in these contact zones, it really suggests that the 4 we've surveyed here are bimodal in nature. And this could be either because the introgression we see is really old and hybridization doesn't happen in present day, or it occurs like present day, but it's rare enough that we either did not sample them or they so quickly backcross with either parental species such that we don't have hybrids mating with hybrids. And we do see this linear spatial relationship between admixture and distance to the other taxon in just one of these sulfur populations. And again, this seems to be due to some potential drift-mediated clustering in the sulfur. But in contrast, we don't see any fine-scale spatial patterns in the outcrosser. And this is likely due to the fact that the scale over which we're sampling within a contact zone is smaller than the scale of pollen flow. So it's likely that even if there is hybridization happening more frequently, like where these taxa come together, that pollinators are moving this sulfur ancestry throughout the population. And lastly, the inclusion of this 4th site, this S6 site with its real high admixture, funky floral types, and chloroplast capture further provide evidence that introgression in this system largely varies at these coarse spatial scales among contact zones. And this is really exciting and continues to open up doors for exploring, the demographic histories of these different sites and how environmental variation among sites might mechanistically promote or hinder introgression. And with that, I also just want to make an exciting plug that next fall I will be starting up a lab at UT Austin. Hook 'em horns, right? This is when Peter's like, everybody clap. Yeah, and so we'll be doing, working on evolutionary ecology and evolutionary genetics of plant adaptation and speciation using a variety of methods. And of course, we'll be recruiting grad students, postdoc, lab techs. I got money. And we also have some really awesome postdoc fellowships within the university too, if you're interested in independent projects. So if you're interested, please get in touch, or if you know folks who might be, send them my way. And with that, I am happy to take any questions. Thank you. Yeah. Thanks. Yeah, so we definitely see structuring within that sulfur, which is to be expected. But yeah, there's not much structuring between outcrosser populations in that contact zone. So one of the things that we're going to be doing moving forward is looking at variation across the genome in introgression and how shared that is among individuals, like within and between populations. So yeah, hopefully next year I'll be able to tell you more about that. Yeah. Okay, thank you.
Thank you all very much for coming. My name is Brooke Kern. I am a PhD student in Dave Moeller's lab at the University of Minnesota, and we are continuing on the Clarkia theme. And I'm going to be talking about how reproductive character displacement reduces opportunities for these taxa to hybridize. So, as many in this room probably are very aware, flowering plants are an incredibly diverse group, and they have radiated very rapidly. And many of these radiations have been driven by pollinator shifts, leading to new floral phenotypes and rapid speciation. However, even though pollinator shifts are very common in angiosperms, an even more common transition that has been less studied in this context is transitions in mating system from outcrossing to primarily self-pollinating, which may be the most common evolutionary transition in angiosperms. And not only is this transition common, but we see very consistent phenotypes arise with this transition, as sulfurs usually have this selfing syndrome, where they have smaller flowers with less separation between the sexual organs, and they also make less pollen. There are quite a few reasons that we might expect a transition in mating system to lead to reproductive isolation. Often sulfurs have faster flowering time. They may move into different habitats. And also, if the selfing flower is so selfing that it is not visited by pollinators at all, then pollen cannot be exchanged between the two taxa. We also might expect to see some post-mating barriers. Just this rapid divergence could cause intrinsic crossing barriers. And we also might expect hybrids to be selected against because their intermediate flowers may be worse at both selfing and outcrossing than the parents. Even though all of these seem logical, there's actually been very little empirical work examining the role in mating system shifts in increasing reproductive isolation, and whether this is an important pathway for speciation remains an open question. So for my talk today, I'm going to be focusing on this idea of floral divergence and reduced pollinator visitation driving reproductive isolation. My study system for this is Clarkia xanthina. This is a pair of taxa currently classified as subspecies, which differ in mating system, with subspecies, do I have a pointer? Yeah, subspecies xanthina is the outcrosser, and then subspecies parviflora is the sulfur. These diverged quite recently, and they have strong reproductive isolation, but it's mainly in pre-mating barriers, so we can still easily cross these in the greenhouse. And as we just learned, there is history of introgression between them, which varies between different contact zones. I want to quickly highlight the floral phenotypes that I'm interested in. So the first one is quite obvious. The outcrosser is bigger. In addition, it has a lot more space between the anthers and stigma, and the stigma is not mature when the flower first opens, so there's also separation in time between the anthers and stigma. In contrast, in the sulfur, you can see that the anthers and stigma are all right next to each other, and the stigma usually opens within a day of the flower opening. These subspecies are parapatric with this narrow contact zone. This is a zone of secondary contact with the oldest parviflora populations being in this far eastern part of the range. And we see this very interesting pattern in phenotypes when we look across the range of these two subspecies. There is a strong pattern of reproductive character displacement. If you compare an outcrosser and sulfur in the contact zone, they are very different in flower size. But if you compare an allopatric sulfur and an allopatric outcrosser, that difference is much less pronounced. And you can see this pattern is stronger in the sulfur, parviflora, so I'm going to be focusing on parviflora for the remainder of this talk. We do have evidence from prior work that this character displacement reduces hybrid formation in parviflora, supporting that this could have evolved due to reinforcement. That was done by placing allopatric phenotypes in the contact zone, and we saw that they hybridized more. So we see this pattern in flower size, but we don't see it in other mating system traits. So this is that same data, just with actual numbers this time, but if you compare that pattern to hercogamy and protandry, we don't really see this trend like we do with flower size. So this led logically to the question of why we only see this trend in flower size. And my hypothesis is that it's because flower size is the mating system trait that's important for affecting pollinator visitation. So which floral traits do reduce pollinator visitation to these sympatric parviflora populations? This question could be hard to investigate because floral traits are usually really highly correlated in natural populations. So we generated recombinant inbred lines of parviflora to test this. These were created by crossing a sympatric line and an allopatric line. We also see polymorphism in color in many parviflora populations, so we incorporated that into our cross as well. So these lines were crossed to make F1s and then selfed to make F2s with recombination. And then we selfed those F2s for 6 more generations to create the lines. And you can see in these images that these lines have diverse phenotypes with the trait correlations somewhat broken up. Since we had these lines, we also did QTL mapping to see if these floral traits were co-localized, and they definitely are. We see a lot of co-localization of all of these mating system traits, which makes sense because our RILs had still had more correlation than we expected. But despite this, we still see a wide variety of phenotypes and are able to test the role of each phenotype individually. And this also makes the pattern of only flower size changing across the range more surprising because these traits are all correlated with each other. So to test how each trait affected pollinator visitation, I set up potted plant arrays of the parviflora RILs in natural populations of the outcrosser. And I did this in 2 contact zone populations for a total of 227 plants, and we were able to do a total of 73.5 hours of pollinator observations. So we saw about 2,000 visits, but despite this, the data are very zero-inflated, which makes sense because we're measuring pollinator visitation to a sulfur. So to accommodate this, we fit a zero-inflated hurdle model, which basically has 2 phases. The first phase looks at which traits affect whether a plant received 0 visits or at least 1 visit in a given observation period. So this is similar to a binomial model. And then the second phase removes the zeros from the data set so we can fit a normal GLM and see how the traits impact the total number of visits in an observation period. So for the first part of that model, we found that the pollinators preferred to visit pink flowers to white flowers. And we also found a significant effect of petal area. So the plants that had larger flowers were more likely to receive at least 1 pollinator visit in an observation period. And then for the second part of the model, we again found that petal area was significant, with larger plants receiving more visits in a period than smaller flowered plants. We did not see a significant effect of our other 2 mating system traits, so this data support our hypothesis that the reason we see this character displacement in petal size, but not in other selfing traits, is because petal size has been selected against in the contact zone to prevent hybridization with the outcrosser. Of course, this is not definitive proof that petal size prevents pollinator visitation, or prevents hybridization. So to follow up on this, I am working now on determining the proportion of the offspring of these same field plants that are hybrids with Xanthina. We expect that these same traits that affected pollinator visitation will also affect hybridization rate. So I should have those data soon, so stay tuned. That is everything. I talked very fast. So yeah, I would like to thank everybody who helped with this project. This was a huge amount of labor because we had to drive the potted plants to California and then set them up in field populations and do all of these pollinator observations. So I especially want to thank my field technicians who were amazing. And I would love to take some questions. the question was about post-pollination barriers. We do see lower seed set in crosses than in parent-by-parent crosses, but it's not a huge difference, so it definitely wouldn't alone prevent hybrids or cause super strong selection. I suspect that there may be some extrinsic hybrid isolation, and that's another chapter of my dissertation. I did an experiment placing F1s in the field to see if they were able to attract pollinators or self or both, but I don't have those results yet. But the preliminary results show that they are intermediate in ability to attract pollinators. So if they're also intermediate in ability to self, that could be a source of selection. Yes?
That's really fun. Thanks. I'm wondering, in your QTL mapping, are any of those regions where all of those traits kind of pile up, are those inversions, or do you think there's some phenotype going on?
I don't know. We got these data very recently, so we haven't really dug into it yet. So yeah, I'm not really sure. We do think we have talked about the possibility that they're inversions, but I think it could also be pleiotropy. And there was also some regions that had lower recombination, so that could be affecting those results as well. Yes. Thank you.
I wondered if you think that there is any kind of decrease in fitness or anything for the selfers who have the bigger flowers and are attracting pollinators in the contact of them. Is there a reason for the selfer that would be selected?
Yeah, so whether there would be a reason for selection against large flowers other than the outcrosser presence. Yeah, I'm not sure. I think even the larger flowered ones are mainly selfing, so they have the ability to attract pollinators, and there's some evidence that they might have slightly more capacity to outcross, but they still aren't outcrossing extensively. So I think that they're still able to self with the larger flowers. Probably they self before pollinators are able to visit them. But there could be selection against large flowers just as a resource cost. It might be better to save water by making smaller flowers. But yeah, I'm not sure. Yes?
Yeah, so sort of similar question. Do you plan to get country data for just seed set from these plants? If you have seeds that you can look at to assess?
Yeah, we weren't planning to get fecundity data because we had some problems with the plants because of heat stress. So I suspect with the potted plants that there were other things affecting fecundity that I would be concerned about interfering with that. But it would be really interesting to see if the ones that were larger flowered and maybe had more hercogamy were less fit because they couldn't self. Yes?
That was a great talk.
Thank you.
I'm curious if you were able to score the pollinator identities at all, and decided if
Yes, and I get to use one of my secret extra slides. Yeah, so most of the pollinator visits were these Clarkia specialists, which is mostly what we see visit the outcrosser in the contact zone, so that's kind of what we expected. So they were mostly this Hesperipus regularis, and then also a lot of these two Megachile species that we aren't able to distinguish in the field. Yeah, there were only about 200 generalist visits out of the 2,000, so it was really dominated by the Clarkia specialists.
Thanks everybody for coming. My name is Keyi. I'm a PhD student in University of Michigan, and I'm happy to present to you all today about mountain radiations in the plant order Ericales. So it doesn't work that way. Okay. So as all of you probably already know, that there are lots of studies that found radiations of plants on mountains. So basically, a lot of them found a shift from lowland to highland often coincide with a shift with higher diversification rate. For example, lupinus is a very famous example. And then other studies have found the same pattern in different clades, different mountain ranges, and then also mountains uplifted at a different time. For example, this is an example of a very old mountain. So it seems like there's no boundary to this phenomenon. It seems very broad and very common. But then people have found counterexamples. They have not found a shift to faster diversification rate in other clades. So then I wonder, how general is this pattern? Then before I go into my method, I want to talk about why people think mountain distribution can lead to faster speciation rate. The main hypothesis is the altitudinal climatic gradient. So people think that this will create a diverse niche for species to diversify and coexist. And also it can create sky islands that serve as geographical isolation that promote speciation. And for the rest of the talk, when I talk about rates, it's going to be tip speciation rate for your information. So my specific question is whether there's a positive correlation between distributions on mountains and tip speciation rate. And different from previous studies are all case studies. My study is going to be more broad across a big clade of plants, which is Ericalis. Why Ericalis is good for this kind of question? Because it has many independent instances of shifting from lowland to highland, and also there's no examples of high species richness on mountains, for example, rhododendron. And it's a floral diverse clade and economically important. For example, persimmon, kiwi, tea, Brazil nut, and blueberry are all in this order. It has 22 families and about 12,000 species. The tree we are using from Carruthers et al. Has about 40,000 species that have occurrence data. So I use BAM and DR statistics to estimate tip speciation rate. I'm sure everybody knows BAM, and DR is just a function of tip branch lengths. I used it because it's a very fast method for big trees. So DR and BAM are highly correlated, but BAM is very conservative in terms of estimation, and DR are more variable, but there can be spurious variation because it's very simple calculation from the phylogeny. I know clades are the better way out, but I couldn't get clades results in before this talk. So I have two measures for distributions on mountains. First, I call it the M index. It's basically just a percentage of species distribution on mountains. So I take the occurrence data and then combine with map of mountain ranges across the globe, and that will give us the percentage of the species distribution on mountains, which for this T species is 21%. And then the other measure is about climatic variation, because we're interested in the altitudinal diversity, climatic gradient on mountains. So it's characterizing distribution in climatic variable regions on mountains. So I took 5 bioclimate variables. 2 is about temperature, 2 is about precipitation, 1 is growing season length. So originally, the variable is about 30 arcsecond at resolution. So I take every 5 times 5 grid cells and then calculate the maximum difference of this variable, and then turn this 5 times 5 grid cells into one giant cell, and then put the maximum difference there. So that will give me a map of a lower resolution, but then it will reflect the climatic variation in that region. And those are the result variables on the map. So the lighter colors means that there's higher variation in this variable. And I combine all of those into one single plot and only keep the mountain regions on this map. It might be a little bit hard to see, so I zoomed in a little bit. You can see the lighter colors are higher in terms of variation in climate. So then, similarly, I combine it with occurrence data, and then it will give us the MCV index, which is abbreviation for mountain climatic variation. For this T species is 2.01. So basically, higher number means that it's mostly distributed in climatically variable regions on mountains. So you can see that this species have 21% distribution on mountains, but it has a rather low MCV index. So let's look at some results. This is the plot for M index. There's lots of variation across Ericales, and red means high, blue means low. I pointed at a few clades that have lots of distributions on mountains. For example, rhododendron, primulaceae, the primrose family, Polymoniaceae is the flax family, and Ciaceae is the tea family. So if we look at variation across different taxonomic scale, most of the variation is below genus level, which is the residual rule. And then if we combine the M index and the MCV index in the same plot, so the inner tree is the MCV index, and then the outer ring is the M index, you can see that there less variation below the genus level for the MCV index, although the trend of colors kind of match. So if we see how correlated are these two indices, they're highly correlated, but the M index is only explaining a very small fraction of the variation in the MCV index, which means if you live on a mountain, it doesn't mean that you live in climatic variable regions, which makes sense. And if we look at BAM results, like I said, BAM is very conservative. So you can see like big blocks of rate regimes on the tree. And most of the variations above genus level, which is kind of not what we expect. Distributions on mountains is going to explain this kind of variation. So BAM is not a very good variable for tip rates for my study. And if we look at DR, there's more variation below the genus level. And then if we see the correlation between all of those indices, BAM, like what we expect, is not correlating with anything. And most of the indices highly correlate with DR, but then it's only explaining a very small fraction of tip speciation rate. So if we put those numbers into trees, like before, the inner tree is the MCV index, and then the outer tree, this time, is the DR statistics. You can see the coloration kind of match. So you can kind of see the correlation over here. But if we change it to BAM, so this time the outer circle is BAM, and the colors doesn't really match this time. So then because there's not very much variation explained by the mountain indices, so I divided this tree into 13 chunks, and then see if those correlate with DR in the specific clades. And only a few clades are significant. Sorry. Only 5 clades I have found that the indices are significantly correlating with DR statistics, and at the same time, a lot of them are not significant with both indices, which is interesting. But the good side is that more variation in terms of speciation rate is explained by these variables in the individual clades. So if we take a look, so for example, for this is several genera in Ericaceae. The MCV index is explaining 21% of the variation. And for the T family, M index is explaining 31% of the variation. So if we look at the trees for those, the left side is TR statistics, and the right side is M index. And you can see the correlation pretty clearly with the coloration over here. And also for the genera in Ericaceae, the trend of coloration matched pretty well. But I still have doubts for these results because, like I said, DR might have spurious variation. So the way out will be have class results and see if it confirm my findings with other parameters. So, in conclusion, indices for mountain distribution only explains a very small fraction of variation in tip rates across Ericaceae, and the effect of the indices is very clade specific. So trying to correlate across a big clade of plants doesn't really work. And also, if you have high distribution on mountains, it doesn't really mean that you'll automatically have higher speciation rate in this clade. So for future steps, I would love to get class results back and then add more bioclimate variables, which I doubt is going to change very much. And then I would like to include temporal climatic variation variable because the variable that I had is geographical variation. But during glacial cycles, the temporal variation of climate on mountains also promotes speciation rate. So by that, I want to acknowledge the Smith Lab, and then special thanks to Thais and Matthias for the help on this project. Yes.
Do you plan on within Ericaceae doing any like geographic or like demographic modeling at all to see whether the occurrence on mountains is coinciding with speciation or whether they speciated and like got to the mountains later, and if that sort of difference in history could contribute to the different kind of unexplained variation you're seeing in speciation rates with mountain dwellers?
That's a very good question. I was thinking about like maybe other factors is influencing the different patterns we see in different clades. For example, on different mountains, age, and the reason that you're talking about. But I don't see very many clades correlating with the mountain indices, so I don't really know if I can find a pattern with not very many examples. So, but yeah, that's something I've been thinking about.